Good morning, students. Today, we will have the introduction about the Unit 2, which exclusively uh, discusses the DNA replication, mechanism of DNA replication in prokaryotes, mechanism of DNA replication in eukaryotes, DNA recombination, so on and so forth. So molecular basis of DNA replication, we will be discussing in the coming lectures. So as far as Unit 1 is concerned, we have covered all the aspects except one topic, that is matrix proteins, that I will discuss later on. And the reason is something. I just want to clear this now first, then I will switch to the matrix proteins. If you see the syllabus, you will see matrix proteins I haven't discussed yet. That is the only topic in unit number 1. So coming to the unit number 2, it deals with the DNA replication. Okay. If I ask you, who gave the 3D double-stranded structure of DNA? It was Watson and Crick. I guess everybody knows this. I hope even you guys know this. He was awarded Nobel Prize for the same. Watson and Crick, they proposed in 1953 that a DNA double-stranded structure looks like this. If you remember, it is like this. Like double-stranded. See, these are the bases. Do you know this? Helical structure. Do you have the prior information of this? Please confirm. Do you know that the DNA is a double-stranded helical structure? This is the DNA. In this shape, your cell contains the DNA, double helical, double strand. This is one strand. Sorry. I hope you guys know that DNA is a double helical structure. This is when you synthesize a strand, it is like this. You synthesize a strand, it is like this. These two strands join. The bases join. I have discussed this in the previous lecture. Adenine with the cytosine and guanine with the AT, G and C. Guanine with cytosine, adenine with thymine, right? And once these two strands, this is a single strand, this is one more strand. These two strands join together. The bases make the hydrogen bonds, and then they make a helical like this. They make a helical like this. And these are the bases. And outside is phosphate and sugar. Did you guys know this? Yes or no? Please tell me. आप लोगों को पता था कि डीएनए डबल स्ट्रक्चर वाटसन और क्रिक ने दिया था 1953 में? तो लोगों को पता था क्या ये? Am I audible?
Yes, sir.
Yes, sir.
Yes, sir.
You knew it, right? Fine.
Yes, sir.
Thank you. Now, आप लोगों ने एक और नाम सुना होगा that we call as template. What is a template? Template means format. If I have to give the meaning of template in simple English language, it means format. Format. See, template, what do you mean by template? Format. If I have to ask you, please write an application, you will ask yourself, what is the template for that? Is there any specific template for the format to write the application? It's a format. आपको कैसे application लिखना है? अगर आपको, if you have to prepare your practical journals, you ask me the template format. Precisely in DNA, we have a template. Can anybody tell me what is template in DNA? Yes, please. Anybody? Does anybody know the meaning of template in molecular biology? Yes or no? Okay, I will tell you.
Sir, okay.
Rishabh, tell me, what do you know
So
about?
one side of DNA, nitrogenous basis.
Sorry, say again.
Sir, nitrogenous basis, one part of DNA can be used as a template.
हाँ, template में कितने strands होते हैं? One strand or two strand? DNA is a double-stranded structure, right?
One
DNA is...
strand.
Okay, fine, good, good, good. Now look here. I am trying to explain you the basics of DNA replication. Before I go into the replication mechanism, you should have something clear. See, if this is a double-stranded DNA, okay, this is double-stranded DNA. This is double-stranded. This is one. This is two. This is normal DNA. Before the DNA replicates, these two strands are coiled together, twisted together. These two strands have uncoiled. When they uncoil, they become separate strands, single strands. And this single strand is called as template. This single strand is called as template. The single strand of the DNA is called as template. And what happens to this template? This template will synthesize similar one strand. This template will synthesize similar one strand. Okay. You will get to know it, how these single strands synthesize the DNA. That we get to know. But as of now, you have to understand that template means template is a single stranded DNA, which facilitates the synthesis of one more strand. If this is a DNA, if I say this is a template, that means this is a single strand, and it will facilitate the synthesis of one more DNA like this. That we call as template. Okay. Now, what I am telling is what I have written here. As you know, the complementary nature of two nucleotide strands in DNA. That complementary nature means what? This is one DNA strand, this is second DNA strand. This A always joins with T. G always joins with C. Right? This is complementary nature. These are the complementary base pairs. Okay. That during replication, each strand can serve as a template for the synthesis of new strand. When these dissociate, when these strands separate each other, each strand serves as a template. Template means format. Because from this strand, one more strand will synthesize. From this strand, one more strand will synthesize. New strand synthesize. This is the basics of DNA replication. And the single strand is called as template. Remember that. Now coming to here. One more thing, whether DNA replication is conservative, semi-conservative, or dispersive. I say, here we say, do you know anything about this? What you have heard about DNA replication? People talk it is semi-conservative. Some people say it is conservative. Some people say it is dispersive. What do you know about the DNA replication? Is it conservative or semi-conservative? DNA replication is semi-conservative. What do you mean by semi-conservative nature? We will get to understand this in coming lectures. DNA replication is semi-conservative. Remember that. Semi-conservative means half of the DNA is found in the new DNA. Half of the DNA is found in the new DNA. Half of this is conserved, okay? In each replication, half. Initially, scientists gave three models of DNA replication. Some people thought that it is conservative in nature. DNA is conservative in nature. Some people thought that it is dispersive in nature. Some people thought that it is semi-conservative in nature. Later on, it was proved that DNA replication is semi-conservative in nature. So before that, we have to discuss these three models of DNA replication. So the first model is conservative model of DNA replication. I will show you the diagram first. You see, look at the diagram, please. If you understand the diagram, no, it will be very easy for you to understand the rest of the things. See, this is your first DNA, mother DNA, right? Two strands, one strand and two strands. What this conservative model says that when these two strands separate each other, these two strands separate each other, and as I said, each strand serves as a template. Okay, this will synthesize this template, new strand DNA, and this will synthesize new DNA. So this is one. Two. After each strand synthesizes the new DNA strand, what happens? These original strands join together again. You see here. And the newly synthesized strands join here again. So now tell me, your 100% old mother DNA has been conserved. In the new DNA strand, double strand, there is 0% of original DNA. Am I right? I will explain this again. See, you have to understand what is conservative model of DNA replication. Conservative model means that the newly synthesized DNA does not contain any mother DNA. See, this is your mother DNA. This is the old DNA. This is DNA strand. This one. This is the strand of DNA, double-stranded DNA. Before replication, what will happen? It will unwind. Okay, this will unwind in two strands. This is the mechanism of DNA replication. Now see, this is DNA. This is one strand. This is two strand. Now what happens? Each strand, this one. Each strand will this one. Each strand will give rise to one new strand. See, this will give rise to this strand. And this will give rise to this strand. Now, after the synthesis is complete, what happens? These original strands join together again. They again make the double-stranded DNA and goes like here. And these newly synthesized strands, they join and they make the double-stranded DNA. Now the question is, if you look at this newly double-stranded DNA, it does not contain any part from the original DNA. Does it contain any part? Does it contain any part of the original DNA? Any strand? There are two strands. Both strands are new. No strand is old. No strand is old. And both the old strands, this one and this one, are here. This is called as conservative model of DNA replication because 100% of the old DNA is conserved again. First, it is separated, uncoiled, then these two carry the synthesis. Okay, then again they are coiled with each other. Therefore, you have the 100% original DNA again back, and you have the complete double-stranded, newly synthesized DNA, which does not contain any part, any strand from the mother DNA. You get it? Now look here. Again, this is original DNA. This is new DNA. Now this will undergo synthesis, this part. This again has the original DNA 100% and this new DNA. This is conservative model. तुम्हाला समजलं का conservative model म्हणजे काय होता?
Yes, sir.
Yes, sir.
This newly synthesized strand does not contain any strand from the old DNA. Old DNA पूरा का पूरा 100% conserve हो जाता है. This is called as conservative model of DNA replication. This model says DNA replication is conservative. लेकिन हमको पता है कि DNA is semi-conservative. ये प्रूव कैसे हुआ वो हम आगे जाके देखेंगे. Another one is dispersive model. I will leave this. I will go to dispersive model. What is dispersive model? See, again, this is a coiled DNA. Before replication, what will happen? This will uncoil. Two strands will uncoil. Dispersive model का क्या बोलता है कि ये जो strand है, this single stranded template, it breaks into fragments. ये छोटे-छोटे fragments में break हो जाता है. Similarly, this strand breaks into small fragments और हर एक small fragment new fragment बनाता है. each small fragment makes the new fragment. एक बार सिंथेसिस कंप्लीट हो गई, ये जो जितने भी फ्रैगमेंट्स है रैंडमली मिक्स हो जाते हैं और हमारे पास नया स्ट्रैंड आ जाता है. सॉरी वेट, हमारे पास नया स्ट्रैंड आ जाता है. तो ये भी आ गया, ये भी आ गया. तो डिस्पर्सिव है. ये जो आपका ओरिजिनल डीएनए है इसके कुछ सेगमेंट्स नए में भी हो गए और न्यूली सिंथेसाइज्ड जो है उसके कुछ फ्रेगमेंट्स इसमें भी आ गए. यू कैन सी हियर न्यू सिंथेसिस फ्रेगमेंट्स. सो डिस्पर्सिव मींस ये जो पुराना अपना जो मदर डीएनए है, जो टेम्पलेट है, it breaks into small fragments and each fragment synthesizes a new DNA. After the synthesis is over, the fragments from this template and the fragments of newly synthesized strand randomly join together and they make a DNA, double-stranded DNA. That's why it's called as dispersive. And if you look at this, you will see here there's a small fragment from the new DNA, small fragment from the old DNA. See, fragmentation is there. It's called dispersive. This is called a dispersive model of DNA. Is that clear? What is dispersive model? समझ लगा dispersive model?
Yes, sir.
Yes, sir.
Now, the most important model is semi-conservative. और आपको भी पता है, मुझे भी पता है, सबको पता है कि DNA replication semi-conservative है. So what do you mean by semi-conservative model of DNA replication? You see, but this is the semi-conservative model. ये जो DNA है, डबल स्ट्रैंडेड DNA, it will break into, ये अनकॉइल हो जाएगा, इसके दो स्ट्रैंड्स बनेंगे, राइट, मदर स्ट्रैंड्स. ये टेम्पलेट 1, ये टेम्पलेट 2, ठीक. 5. Now this will make one template. For example, इसने रेड बनाया, ये भी एक टेम्पलेट बनाया, इसने रेड बनाया. अब this is a single strand, this is a single strand, right? They have to make a double strand. What happens in double-stranded semi-conservative nature? This is 1, this is 2. This is 1. This is 2. मुझे बताइए इसमें कितना परसेंट ओल्ड डीएनए है? इसमें कितना परसेंट ओल्ड डीएनए है? 50% है ना. One strand is from the old DNA. Am I right? One strand is from the older DNA. There is one new strand and one older strand. तो ये मॉडल में क्या बोलता है? ये मॉडल बोलता है ये जो एक स्ट्रैंड आपकी है, सॉरी आई विल जस्ट, ये आपका डीएनए है. बिफोर एप्लीकेशन इट विल अनकॉइल, इट विल अनकॉइल. तो फिर क्या होगा? अब ये एक स्ट्रैंड टेम्पलेट हो जाएगा, ये टेम्पलेट स्ट्रैंड एक नया स्ट्रैंड बनाएगा और ये भी एक नई स्ट्रैंड बनाएगी. तो फिर क्या होगा? ये जो ओल्ड स्ट्रैंड है ये नई स्ट्रैंड के साथ ही ज्वाइन हो जाएगी. ओल्ड स्ट्रैंड ज्वाइन होगी न्यू स्ट्रैंड. ये जो तुम्हारे पास हेलिकल स्ट्रक्चर था, अब तुम्हारे पास दो हेलिकल स्ट्रक्चर आ गए. नाउ यू हैव टू हेलिकल स्ट्रक्चर्स, राइट? And each helical structure contains one old strand and one new strand. One old strand and one new strand. That is called as semi-conservative nature. हर दूसरे replication में, हर दूसरे helical double helix में एक strand old होगी, एक strand new होगी. And half of that is called semi-conservative. Half of the old strand goes to the new double helical structure of DNA. You see, now look at this. Now look at this. This is a double structure. This is old one, right? And there is one more strand. This is new. Now coming to this is old one. It has come from here, and this one is new. So how much is here new? 50. How much is old? 50. Semi-conservative. Half of the DNA in this is old DNA. It is template DNA. और ये जो नया बना है नया DNA old DNA से बनता है, template से बनता है. And the new strand and the template strand then join together. What happens in conservative model. Once the template joins, you see, once the template, once the here, once the template synthesizes the new strand, the template goes back and joins with another one, joins with the older one. Here, the template and the newly synthesized does not join together. You see, in this, you both have the original templates, and here you both have the newly synthesized strands. So this is why it is called as semi-conservative. अगर आप किसी भी replication model में देखोगे, उसमें जो नया DNA होगा, एक strand, double strand जो होगा, double strand में एक strand new synthesized होगी, and the second will be the template, which will be the older DNA. समझ लो क्या? यहाँ मैं एक बार फिर से explain करूँ इसको. Just look at this. Have a very proper, clear look at the slide. Semi-conservative means half of the DNA is conserved. Look at this. This half comes from here. And in this, the half comes from here. And this is a new one. Because you do not see this here somewhere. ये कहीं नहीं है मदर में. It is not here, right? This is semi-conservative. Now coming down to this. Now look at this. See, there are, this is 1, this is 2. Look, this is here, 50, and this is here, 50. This is here, double strand. Only this is here, and in this is here. So 50. 50% conserve hota hai, because in this double-stranded DNA, 50% is from the older DNA, which is template, and the 50 and another strand is the newly synthesized strand. That's why it is called as semi-conservative nature. And we all know the DNA replication is semi-conservative in nature. This was rejected, conservative model, and dispersive model was also rejected. Now the big question is how the scientists came to know that DNA replication is semi-conservative in nature. How? That is the $1,000,000 question. What is the logic? What is the experimentation? Who is the person behind the observation that DNA replication is semi-conservative in nature? ये समझ में आ गया तुम लोगों को? Did you understand this semi-conservative model? Dispersive model,
Yes, sir.
and conservative model?
Yes, sir.
I hope this is clear. Now we will go to the experimentation. Who is the कौन मानुस था वो जिसने ये सब बताया हमको? Who is the person responsible for making it clear that DNA replication is conservative? Can you name, please? If you know anybody, please tell me. Who is the person responsible for this? Yes, please, please. Yes or no? Do you know? Yes or no? You can message me also. Huh? किसी को नहीं पता है क्या? This is usually taught in the TUI, molecular biology, in the 5th semester, molecular biology. Okay. The scientist behind this observation is, there are two scientists. These are the models I have given the description. Okay. You can see here, conservative replication model, dispersive replication model, and just a minute. Sorry. So this is semi-conservative, conservative, and dispersive model, right? Now coming to this one, yes. So who is the person behind this? The person behind this great work is Michelson and Stock. These are the two scientists who in 1957 demonstrated that the DNA replication is semi-conservative. How? You please listen very carefully few things about this experiment. They have to do the DNA replication. They have to carry out DNA replication to demonstrate the DNA replication is semi-conservative, right? They have to carry out the DNA replication. So what they did, they used the microorganism E. coli in their experiment. Remember that, E. coli. What? E. coli. E. coli. Sometimes the question is asked, name the organism used by Michelson and Stoll in their experiment. It was E. coli. What they did, they used E. coli. You see, E. coli is a bacterium. Okay, it's a bacterium. You can grow the bacterium on nutrient medium, like you can grow the cells, plant tissues on nutrient medium. You can grow the animal cells on the cell culture medium. Precisely, you have the culture medium and nutrient medium for bacteria also. So what they did, they grew E. coli on a nutrient medium containing nitrogen 14 and nitrogen 15. First, they cultured the E. coli on a medium containing nitrogen 15 isotope. Now, as you know, the DNA contains nitrogenous base, right? And the nitrogenous bases contain nitrogen atom. When the E. coli is grown on a specific medium which contains a specific isotope of the DNA, if you extract that DNA from the microorganism, that DNA will contain your isotope. That DNA will contain this isotope because you have provided only this isotope for the growth. So what these scientists did, they carried out the DNA replication procedure of E. coli. And first what they did is, they cultured the E. coli on a nitrogen containing nitrogen 15 and then extracted the DNA. And what they found, that DNA contains nitrogen 15. Okay. This was culture 1. Now, this culture was now transferred to the medium which contains nitrogen 14. This contains nitrogen 14. And after the growth, DNA was extracted. And what they found in the DNA being semi-conservative, what they found nitrogen 14 also here and nitrogen 15 also here. Because जब उन्होंने ट्रांसफर किया DNA को वहां पे, माइक्रोब को वहां पे ट्रांसफर किया, तो माइक्रोब ने वहां रेप्लिकेशन किया होगा ना, जाहिर सी बात है, ग्रोथ के बाद रेप्लिकेशन हुआ होगा. तो टेम्पलेट क्या था? 15, क्योंकि यहाँ से उन्होंने माइक्रोब लिया था. 15 का माइक्रोब उन्होंने लिया था, and that was cultured on nitrogen 14. And when after the growth, what they did, when they extract the DNA, they found the DNA contains nitrogen 14 also and 15 also in equal proportion. That means semi-conservative. Again, what they did, this 14 was transferred to another culture. And then DNA was extracted. What they found 14, 14, and 15. Because now the previous culture contains 14 and before that contains 14. So now the 15 is decreasing and 14 is increasing. Again, third, fourth culture, it contains 14, 14, 14, and 15. Continuously they found 15 is always there, but its number is decreasing. Its number is decreasing. And the 14's number is increasing because every culture is now carried in nitrogen 14. So they found that, yes, this is semi-conservative in nature. Because from the very first DNA extraction, what they found that it was 14 and 15 in equal proportion. This is the DNA. Right? Then again it will come. Again, it was cultured on nitrogen 14. But the part of the 15 is always there. The part. Again, it's continued going to 14. The part of the 15 is always there. What will happen? 14 you will get prominently, but 15 will continue to decrease because you are not providing 15 anymore, nitrogen 15 anymore. Right? So this is how they demonstrated that DNA replication is semi-conservative nature. And the isotope they used is nitrogen 15 and nitrogen 14. But first culture was carried out on a nutrient medium, which was supplemented with nitrogen 15 isotope, so that we can identify the nature of the nitrogen in the DNA. And as you know, and the pentose sugar is attached to base, and the base contains nitrogen rings. समझ लगा?
Yes, sir.
This is a structure. You see, I will just show you the diagram. Please have a look. First culture. Nitrogen 15. Then centrifugation of DNA. Now this is DNA, heavy DNA, nitrogen 15 DNA is here. Okay. Now this is shifted to, again, medium, which contains nitrogen 14. See, this is DNA extraction. Now see the DNA. This contains 15 and 14, nitrogen 15 and nitrogen 14. Okay. Now come down to next. It was again shifted to nitrogen 14. Now see, this contains 14, complete 14. This contains 114 and 115. Now again, this was shifted to nitrogen 14. See, this is 14, this is 14, this is 14. Both the strands are 14, both the strands are 14, both the strands are 14. Here, one is 15 and one is 14. Because 15, you can see the color. And what he found that, yes, 50% is continuously conserved. That is the reason he concluded that DNA replication is semi-conservative in nature. And these are the bands. These are the bands. This is nitrogen 15 band. This is nitrogen 14 band. He demonstrated it extensively. And what he found that 50%. Next, every time you go to the replication, the 15 is getting. See, here you have a complete 15. Here you have 3 strands 14, 1 15. Here both the strands 14, both are 14, both are 14. Here one is 14 and one is 15. The 15 is decreasing because we are not providing any more nitrogen 15 in the medium. So this was the experiment of Michelson and Stoll. Remember that there are many times few questions are asked. Name the microorganism he used. He used E. coli. He used E. coli first, and he grew the E. coli in a nutrient medium containing nitrogen 15. First culture contained nitrogen 15. Cesium chloride. Then he extracted the DNA in the cesium chloride, centrifuged that, isolated the DNA, and found that it contains both nitrogen 15. And then he took one bacteria from this old culture and then transferred it to nitrogen 14. Second one. This is one generation. Second one. Again, he extracted the DNA. Then again, he transported 14. Cycle 3. In the third cycle, started with the nitrogen 15. Then cycle 1, cycle 2, and cycle 3. All the 3 cycles were in and the medium containing nitrogen 14. And he found that here in the first one, we have 15 complete, and in the second one, you have one strand of 14, one at 15, and finally, gradually, when he goes to the next cycle, what he concluded that this nitrogen 15 goes only in one strand, and all the strands contain nitrogen 14. So thereby he concluded that this is semi-conservative nature. Is that clear? Please unmute your microphone. Please unmute your microphone.
Yes.
So we are done with the lecture. Thank you very much. Please drop your attendance here. Please drop your attendance. घर पे थोड़ा स्टडी कर लो और ये डीएनए रेप्लिकेशन है, ध्यान से लेक्चर को देखा करो। यूट्यूब सॉरी मेरा लेक्चर का वीडियो लेक्चर भी आप रेफर किया करो। और अगर कुछ whenever you feel that हमको we want a revision or something, you want me to re-explain it, तो बिना किसी झिझक के आप मुझे बता सकते हो, राइट? और आई होप कि आप लोगों ने ये सब कुछ अपलोड किया होगा. कीप अपलोडिंग योर असाइनमेंट्स. लास्ट डेट इज 18th CIA 1 का, एंड प्रेजेंटेशन हम 18 से पहले ले लेंगे. आई विल गिव यू द शेड्यूल. आई एक्सपेक्ट यू टू बी रेडी बाय नेक्स्ट सैटरडे. ठीक है?
Yes, sir.
By 15th be ready with the presentation. Okay? Yeah, thank you. अच्छा, ये इम्यूनोलॉजी आपको समझता है क्या? द न्यू मैडम हैज रिसेंटली टीचिंग यू.